Hello guys, welcome to my YouTube channel. Today I will discuss about yeast to hybrid system. Yeast to hybrid system is one of the most useful technique in biotechnology to detect protein-protein interactions. So question arises, why do we need to study protein-protein interactions? First one is the functional annotation of proteins. If we want to detect the cellular pathways or the signaling cascades inside a cell, we must need to know the protein-protein interactions. Second one is the biomarker discovery. If we want to detect the biomarkers which are related to different diseases, we must need to know protein-protein interactions. The third one is the drug discovery and development. Drug discovery is important, very useful, and so we need the protein-protein interactions to discover new drugs. So the discovery process. A group of scientists called named Stanley Field and OKQ Song, they first detect the protein-protein interactions in using the GAL4 transcriptional activator in yeast Saccharomyces cerevisiae in 1989. So the basic principle is that we need the yeast cells or the Saccharomyces cerevisiae for yeast to hybrid system, which has a property that they lacks the activator for the LAC-Z gene, which is a reporter gene. And this reporter gene or the LAC-Z gene, it codes for beta galactosidase. Beta galactosidase is the enzyme which converts lactose to glucose and galactose. It breaks down lactose to glucose and form glucose and galactose. Then it can also convert the X-gal into a chromogenic substance. It produces a blue pigment substance after breaking down X-gal. So the yeast colonies which appear blue, it indicates that the yeast cells are producing the beta galactosidase, and that's why the colonies are blue. So now the GAL4 transcriptional activator requires it has two regions. One is the DNA binding domain and another is the activation domain. So the DNA binding domain for GAL4, it will bind with the DNA, and then activation domain will bind to the DNA binding domain, and it activates the DVD. Then the RNA polymerase will be recruited, and that's why the gene will be expressed. The LAC-Z gene will be expressed because of RNA polymerase recruit. Then we need two types of proteins. One, if we want to detect the interaction between those two proteins, one is called bait and another is prey. So we are now want to detect the interaction between those two proteins. So that's why we need two types of plasmid. One is the bait plasmid and another is called prey plasmid. The bait plasmid, in case of bait plasmid, we are conjugating the activational domain and the bait protein in the plasmid, and in case of prey plasmid, we are conjugating the DNA regions of the DNA binding domain and the prey protein. So now those two plasmids will individually produces their proteins, conjugated proteins, activation domain bait protein and the DVD binding prey conjugated protein. So now we need to transfer those two plasmids into the yeast cell, and those two plasmids will produce their own proteins inside the yeast cell. So suppose if the gene will only express if those two proteins are interact, like if these two proteins, bait and prey proteins, they interact, then only the RNA polymerase will be recruited and then only the gene will be expressed. So now if the gene will be expressed, then beta galactosidase will be produced and it will cleave the X-gal and produces chromogenic substances, blue colored. Now if those two proteins, suppose they are not interacting, in that case RNA polymerase will not be recruited and the reporter gene, LAC-Z gene, will not express. So there may be four chances. The first one is the normal case where where we are not conjugating the bait and prey proteins into the plasmids. So in that case, the reporter gene will be expressed because of the activation domain and the DNA binding region. The second case, if a yeast cell gets only one of the plasmid, then it will only produce one of the proteins, the conjugated proteins. Then it will be not the genes will be not expressed. In second case, if it gets the prey and the activation domain plasmid, then it will also can't express the gene. So the gene expression will only happen if those two plasmids are present inside the yeast cell and those bait and prey proteins are interacting with each other. Then only the reporter gene will be expressed. So now how we can detect it? We can see the blue colonies. These are appeared because of the production of beta galactosidase. Uh, and and white colonies also can produce if those two proteins are not interacting, so that beta galactosidase is not produced. So thank you. Please like, share and subscribe to my YouTube channel.